Welcome. Today's topic it is a genomic library. Whenever you come across the word library, what is in your mind that the all the books they are kept at one place. And when we go to the library here, it is very easier for us to find out a particular book in which you are interested. In the same way, here the concept of the genomic library it is there. Here it is a collection of, okay, here it is a collection of all the DNA sequences which are placed over the genome. That is, here when we are going to construct the genomic library, you are going to go for the fragmentation of the DNA with the help of the restriction endonucleases. Previously, the restriction endonucleases which is used, it is EcoR1. So the restriction digestion of the isolated genomic DNA, it is going to be done. And then the fragments which are obtained, it is going to be cloned into the lambda-based vectors, and then there was a screening for the desired gene. Now, when we are going to do this, why we have to do this? Okay, here it is going to be done to isolate a desired gene. Okay, so whenever we go for the cloning, okay, whenever we go for the cloning, there is a need of, okay, there is a need of the particular gene to be cloned. And that particular gene, when it is that particular gene, now it can be taken from the library, and that library it is called as an genomic library. Now here, whenever we go for, okay, that is the previous approach which has been used. It is the digestion with the EcoR1. This EcoR1 enzyme, now it is allowing cutting of the human genome into the fragments, and the size of that fragment, it is of about the 4 kb fragment. Now here there are the problems, okay, the problems to use the EcoR1 enzyme. First problem, it is going to cut the gene internally. So the number of fragments of the gene, it is going to be present. And here you are not going to isolate a single fragment on which there is a placement of the complete gene. Second one, it is what? That the lambda vector that are going to be used for the library construction. Here the insertional size, okay, insertional size, it is very low. It is of about the 4 kb. That is the capacity of the vector for the lambda. It is higher than the 4 kb. So here it is inconveniently a shorter DNA fragments. And it may happen that the two restriction sites for the EcoR1, they are placed apart, placed far away from each other, so that whenever there is a cutting of the DNA, a large fragment of the EcoR1, it is going to be obtained, okay, and that fragment, it is unable to get accommodated into the vector. Now these are what? These are the number of problems when we are using the EcoR1 enzyme, as it is going to generate the 4 kb of the fragment. So there is, okay, then there is, what is the solution for this problem? Here, the solution for this problem, it is isolate the random DNA fragments. Whenever you are going to isolate the random DNA fragment, the advantage of it is what? It is, it will not systematically, there is no exclusion of any DNA sequence. And here, the clones which are obtained, that they are the overlapping clones. Overlapping means what? This one and this one. That is the last DNA sequences, they are going to overlap to each other. And when these sequences, they are going to be overlap to each other, here we can assemble the genes which are glued in between. Okay. Next here, the size of the DNA fragment, it is a larger one, so that, okay, so that the fewer clones they are required to complete the library. Now here, how to get the random fragments? There are the two ways: use mechanical shearing, or use the enzymes, and that enzyme, it is a restriction endonucleases too. Now the Maniatis, he has given the strategy for producing the genomic library, and in his strategy, he has used the two enzyme, and that two enzyme, it is a He3 and ALU1. So here first the genomic DNA, it is isolated, and now it is going to be cleared with the restriction endonucleases 2. And here the enzyme, it is a He3 and the ALU1, and the digestion, it is a very partial digestion. Here the partial digestion, it is done by, because we are requiring the a fragment which is a larger one, that is it is about the 20 kb of the fragment. For that purpose, there is a use of the digestion, and that digestion, it is a very partial digestion. Now, next here, there is a use of the eco-R1 methylases. And these eco-R1 methylases, they are used to block the eco-R1 sites in the DNA fragment. Now, before the methylation, we have to go for the size fractionation. Size fractionation means what? Now, when we have digested the genomic DNA with the restriction enzymes, the number of fragments they are generated. From that number of fragment, we have to choose a fragment which is having the size of the 20 kb. For that purpose, here we have to go for the agarose gel electrophoresis, in which there is a separation of the DNA fragments. And from that separated DNA fragments, now you are able to pick up the 20 kb fragment. And now that 20 kb fragment, it is treated with the eco-R1 methylases to block the eco-R1 sites. Now here it has been shown that it is the 20 kb fragment, and this 20 kb fragment, now it has been methylated one. Now here, the He3 and the ALU1, both of them, they are going to generate the ends, and that ends, it is called as the blunt end. Okay, and now to this blunt end, there is a ligation of the eco-R1 linkers. Now here, what do you mean by the linker? Linker, it is a short sequence of DNA which is consisting of the restriction site. So here, when we are adding the eco-R1 linker to the 20 kb fragment, actually we are ligating a restriction site to whom? To the 20 kb fragment. Now after that, there is a digestion with the eco-R1 enzyme. So here, the cohesive ends generated by the eco-R1, now it is observed. Now here, as we are going to use the eco-R1 enzyme, this restriction enzyme, to digest the linker molecule, here we have to use this eco-R1 methylases. Now here, the use of the eco-R1 methylases, it is hiding the eco-R1 sites which are present over this 20 kb fragment, so that when we are treating our 20 kb fragment with the eco-R1 enzyme, at that time, this 20 kb fragment, it is going to be a intact one. It is not going to be digested with the eco-R1 enzyme. That's why there is a need of what? There is a need of this eco-R1 methylases to methylate the eco-R1 restriction site. Now here, the vector in which we have to clone this, okay? We have to clone this 20 kb fragment, that vector. Here it is a caron 4A. And this, it is a replacement vector. Now here, and the vector molecule, now it is going to be digested with the eco-R1. So here there is a generation of what? There is a generation of the ends, okay, the cohesive ends which are generated by the eco-R1. And these ends, okay, now they are how? They are complementary, or they are complementary to the ends which are present over this 20 kb fragment. Now here you can see, okay, here you can see your insert molecule. It has been inserted, that is your 20 kb fragment. It has been inserted into where? Inserted into your lambda vector. And next, this DNA molecule, now it is going to be packaged into the phage head by the in vitro packaging. Okay, in this way, the Maniatis, he has given the strategy to do the genomic library construction, in which there is a use of the He3 and the ALU1 restriction enzyme, which are the tetra cutters and which are going to generate the blunt ends. And here, the linker technology, it is used for the ligation purpose. Now, the next one next one here, it is what? There is the modified technique, it is going to be used. The enzymes, they are now going to be used. They are other than the He3 and ALU1. And here the enzyme, okay, here the enzyme, it is a Sau IIIA1 enzyme, and it is also going to generate, okay, it is also going to generate the ends which are which are complementary to the BAM-H1, and both of them, they are going to generate the cohesive ends. Now here, the Sau3A1 fragment can be readily inserted into the lambda replacement vector like the lambda EMBL3, which is digested with the BAM-H1. So when the replacement vector EMBL, it is digested with the BAM-H1, the end generated, they are how the end generated, they are the same like the end generated by the Sau3A1. Okay. Now here we can see, okay, or we can see here, uh, due to this convenience and efficiency of this strategy, the lambda EMBL series vectors have been very widely used for the genomic library construction, because there is a use of the Sau3A1 enzyme which is used for the isolation of the 20 kb fragment, and the Sau3A1 end generated, they are like the BAM-H1, ends, and this BAM-H1, it is allowed to cleave the lambda EMBL3 vectors. Now these lambda EMBL3 vectors, okay, that they carry the red and the GAM genes on their stuffer fragments. Okay, as it is a replacement vector, it is going to replace a stuffer fragment with a insert molecule. Okay, so here the stuffer fragment which is present, it has a genes red and GAM genes. Okay, so whenever there is a replacement, okay, when there is a replacement of the stuffer fragment with the insert, this red and GAM genes, they are going to be deleted from the lambda EMBL vector, and here allowing the ligation of the insert, that is a 20 kb fragment. Now, due to this, okay, there is a positive selection, and the selection, it is done as a SPA phenotype. Okay, so here the screening, it is a very easier for the lambda EMBL vectors. Okay, just observing for the SPA phenotype, okay, you can say that the phages, they are the recombinant or they are not recombinant. Okay, so here the next vectors which are there, they are the lambda 2001, and then there is a lambda dash, lambda fix. Now here it is a map of the lambda dash, and you can see, okay, you can see here, these are the multiple cloning sites. Okay, these are the multiple cloning sites. Okay, and here it is the stuffer fragment, and this stuffer fragment, it is consisting of the red and the GAM genes. Now here, when you look at this lambda dash vector here, okay, this T3 and this T7, okay, this T3 and the T7, these are the RNA polymerases promoters. Okay, these are what? These are the promoters specific for the bacteriophage T3 and T7 for the RNA polymerases. So here, these promoters, they are located here, T3 promoter and the T7 promoter for the RNA polymerases, and you can see it is placed in between the this linker, okay, that is into these multiple cloning sites. Now here's the second one, okay, the second vector, it is a lambda fix vector. Here also you can see there is a multiple cloning sites, they are placed, and in the multiple cloning site there is again the promoter for the T7 and there is a promoter for the T3 RNA polymerases. That is these are the bacteriophage derived promoters which are placed into these vector lambda dash and a lambda fix. And in both of them, there is a stuffer fragment, and that stuffer fragment, it is a red and GAM gene. That both of them, they are the replacement vectors. Now, whenever the 20 kb fragment or the fragment of the genome, it is taken, and at that time, there is a digestion with any one of this enzyme. And here, as we have seen, that when there is a Sau3A1 ends, at that time we are going to cut this vector with the help of this BAM H1. Okay, here it is a BAM H1, here it is a BAM H1. So you are going to cut the vector with the BAM H1. So the cohesive ends, they are going to be generated. This Stuffer fragment, now it is going to be released, and this, instead of this Stuffer fragment, here there will be the insert. Okay, so as there is a loss of the red and gam gene, okay, the vector, it is now allowed to grow into the T4 lysogen. That is, there is a SPA phenotype. Okay, there is a selection, there is a positive selection, and that it is a selection for the SPA phenotype. Okay, and here the specialty of this vector, it is what you can generate the RNA probes as you have. Okay, as here on this vector there are the promoters, T3 promoter and the T7 promoter. On both of them there is a promoters. So when here, if the RNA polymerases for the T3 or the T7 it is used, at that time you can have the RNA probes, because the promoter, it is allowing the start of the transcription, and that transcription, it will go through your fragment which has been cloned. Okay, so in this way, these lambda dash and lambda fix vectors, it is allowing the construction of genomic library, as well as it is allowing the synthesis of the probes for that particular gene which has been inserted in these vectors. Now, next year, for the construction of genomic library, the high capacity vectors, they are used, like the Cosmids, backpack, and the YAC vectors, they are going to be used. Now, what are the advantages of these large capacity vectors? The average size insert, it is larger than the lambda replacement. So the number of recombinants needed to be screened to identify the particular gene of interest is correspondingly lower. Larger genes can be present in the single clone, and fewer clones, they are required to assemble the contig. So in this way, there are the number of advantages of the cloning of the DNA fragment into the high capacity vectors. I think you have cleared the idea about the genomic library. So if you like my video, please share my video and subscribe my channel. Thank you.